Imagine a world where we can create identical, disease-free plants from a tiny piece of tissue. This is the reality made possible by plant tissue culture. In this revolutionary technique, scientists can grow plants from individual cells, tissues, or even organs in a controlled lab environment. Through a process known as totipotency, where a single plant cell can regenerate into a full organism, plant tissue culture is transforming the fields of agriculture, biotechnology, and horticulture. From saving endangered species to creating genetically modified crops, the possibilities are endless. Today, we're going to explore the fascinating world of plant tissue culture and understand its techniques, types, and broad applications. Before diving into the specifics, let's understand the fundamental principle behind plant tissue culture. The concept at the core of tissue culture is totipotency, the ability of a single plant cell to develop into an entire plant. This concept was first discovered in the mid-20th century and has since paved the way for a variety of techniques used in plant science today. Totipotency means that even a tiny fragment of a plant can regenerate into a whole new plant, but it doesn't happen on its own. The process begins with the selection of an appropriate plant part, known as the explant. This could be anything from a leaf, a stem tip, a root tissue, or even a single cell. The selected explant is then sterilized, usually with chemicals like bleach or alcohol, to prevent any microbial contamination. Once sterilized, the explant is placed in a special growth medium, a nutrient-rich gel that contains essential vitamins, sugars, plant hormones, and agar, which helps solidify the medium. The right conditions must be created for the explant to grow and regenerate. Now let's break down the techniques involved in plant tissue culture and how we get from a tiny explant to a fully grown plant. The first step in most tissue culture processes is the induction of callus formation. This is where plant cells divide and form a mass of undifferentiated cells called a callus. This process is promoted by the application of certain plant hormones. For example, auxins help with root formation, while cytokinins encourage shoot development. Once the callus is formed, the next step is organogenesis, the process by which callus cells differentiate into specific plant organs like roots and shoots. By adjusting the hormone ratios in the medium, scientists can control whether the callus develops roots or shoots. This step is essential for producing a complete plant from the callus. Another fascinating technique in tissue culture is somatic embryogenesis. In this process, non-reproductive or somatic cells are induced to form embryos. These embryos can later grow into fully functional plants. Somatic embryogenesis is important for clonal propagation, which allows scientists to create many genetically identical plants. It also plays a big role in genetic modification. After plantlets are formed in the lab, they undergo a process called acclimatization, or hardening off. In this phase, the plants are transferred to a soil-based or similar growing environment. This is when they adjust to external conditions like light, temperature, and humidity, making the transition from a sterile lab environment to the real world. Now let's take a closer look at the different types of plant tissue culture, which are used for specific purposes. The most widely used form of tissue culture is micropropagation, which involves the rapid multiplication of plants. This technique is often used to produce thousands of genetically identical clones in a short period. Micropropagation is particularly useful for mass production of ornamental plants, fruit trees, and plants that are difficult to propagate by traditional means. In callus culture, undifferentiated cells, known as callus, are induced to proliferate and develop into plant organs. This technique is used in genetic modification and somatic embryogenesis and can also be used to produce valuable secondary metabolites. Anther culture and pollen culture involve the cultivation of the male reproductive parts of plants. The goal is to produce haploid plants, which contain only one set of chromosomes. These haploid plants are essential in breeding as they can be used to create genetically uniform lines. Protoplasts are plant cells that have had their cell walls removed. Protoplast culture involves the fusion of protoplasts from different species, creating hybrid plants with traits from both parents. Meristem culture focuses on growing the actively dividing meristematic tissue of plants. This type of culture is particularly important for virus-free propagation, as viruses usually don't infect the meristematic tissue. It's widely used for crops that are prone to viral infections. Finally, in suspension culture, plant cells are grown in a liquid medium. This method is often used to produce secondary metabolites, such as alkaloids and essential oils. Plant tissue culture is far more than a laboratory technique. It has numerous real-world applications that are changing the way we approach agriculture, biotechnology, and conservation. Tissue culture enables the rapid production of genetically identical plants. Micropropagation, for example, is used to produce large quantities of plants with desirable traits in a fraction of the time required by traditional methods. This technique is ideal for mass-producing crops like bananas, strawberries, and many ornamental plants. Plant tissue culture is also a cornerstone of genetic engineering. By using techniques like agrobacterium-mediated transformation or gene guns, scientists can introduce new genes into plant cells. This allows them to create genetically modified crops with improved traits such as better disease resistance or higher yield. Tissue culture, particularly meristem culture, is used to create virus-free plantlets. By culturing the meristematic tissue, which is free of viruses, scientists can regenerate healthy, disease-free plants. This is especially important for crops like bananas, potatoes, and strawberries, which are susceptible to viral diseases. Tissue culture has become an essential tool for conserving rare and endangered plant species. Through in vitro propagation, plants can be multiplied in controlled conditions, which helps preserve their genetic diversity and aids in their reintroduction into natural habitats. Suspension cultures are used to produce valuable secondary metabolites, compounds like alkaloids, flavonoids, and essential oils. These metabolites are used in various industries, from pharmaceuticals to cosmetics to food production. In conclusion, plant tissue culture is a groundbreaking tool that is transforming the way we grow and conserve plants. From producing genetically identical crops to saving endangered species, the potential of tissue culture is limitless. As research and technology continue to advance, plant tissue culture will play an even greater role in shaping the future of agriculture, conservation, and biotechnology. Despite challenges like high initial costs and the need for skilled labor, the benefits far outweigh the hurdles, making plant tissue culture an essential technique for the future.